So for today's deep dive, we're looking at some, truly foundational biological systems, the stuff that keeps you alive, really.
Yeah, the systems that are constantly in this fight against entropy.
Exactly. We're going to tackle two big ones. First, the dual control of your breathing, both neural and chemical. And then we're going to zoom way down to the single cell, to the mechanics of active transport.
The body's tiny, constant uphill battle.
Right. So, okay, let's just jump in. The control center for breathing. It's so much more complex than just, deciding to take a breath.
It really is. It all starts in the brainstem, the medulla, and the pons. This is your respiratory rhythm generator.
Okay.
And you can think of it as having two basic parts. You've got the dorsal respiratory group, or DRG. That sets your fundamental pace, your quiet, at-rest breathing.
The basic inhale-exhale rhythm.
Right. And then there's the central respiratory group, the VRG. That's, think of it like an emergency throttle.
Got it.
It's pretty much inactive until you're sprinting or under stress.
And it provides that powerful exhale.
Exactly. It kicks in when you need it.
But the real surprise for me in the sources was that the primary driver isn't actually the nerves, it's chemistry.
It is. The chemical regulation is by far the most powerful influence on your breathing rate and depth.
So how does that system work? What's it sensing?
Well, it's sensing CO2.
Okay.
But, and this is the fascinating part, it does so indirectly.
Yeah.
The central chemoreceptors in the brainstem, they don't actually measure blood CO2.
Okay.
Instead, CO2 diffuses from the blood into the cerebrospinal fluid, the CSF, where it immediately forms hydrogen ions, H+ Bra.
Which is just acid.
It's acid. So your brain is actually sensing the pH, the acidity of its own environment. High acid means high CO2, and the brain says, okay, breathe harder, breathe faster, blow it off.
So the peripheral chemoreceptors, the ones in the carotid and aortic bodies, those are just the backup alarm then?
They're absolutely the backup, yeah. They're primarily sensitive to a really big drop in oxygen, like below 60 millimeters of mercury. It's an emergency signal.
And when that whole feedback system gets out of sync, that's when you see something like Cheyne-Stokes breathing.
Exactly. That strange pattern of breathing that gets deeper, then shallower, then just stops for a moment.
Yeah.
Apnea.
Yeah.
It's often caused by circulatory delay, like in severe heart failure.
Mm-hmm.
The CO2 signal just takes too long to get from the lungs to the brain, and the feedback loop starts to oscillate wildly.
Okay, so let's connect that idea of constant uphill regulation down to the smallest possible scale, the single cell.
From the whole body to the cell membrane.
Yeah, we're talking about active transport.
Which is that uphill battle we mentioned. Anytime a cell needs to move a molecule or an ion against its gradient from low concentration to high, it has to spend energy.
Metabolic energy.
Yeah.
ATP. So primary active transport uses that ATP directly. The classic textbook example is the sodium-potassium pump.
The NAK pump, yeah. It's absolutely fundamental. This little protein is constantly burning ATP to actively push 3 sodium ions out of the cell.
While bringing 2 potassium ions in.
And that one action maintains the crucial electrical and chemical gradients for pretty much everything a cell needs to do. It's literally the cellular battery.
And that battery, that low sodium concentration inside the cell, that's what powers everything else, which is secondary active transport.
Precisely. The secondary transporter doesn't burn ATP itself, it just harnesses the potential energy of all that sodium wanting to rush back into the cell. It piggybacks on it.
Right, and we see it in 2 forms. There's co-transport, where both substances move in the same direction, like the NOD-glucose transporter in the kidney.
SGLT, yeah. It drags glucose back into the body against its gradient.
And then the opposite, counter-transport.
Antiport, right, where the energy of sodium flowing in is used to push something else, like calcium, out of the cell, which is vital for heart muscle relaxation.
And when you look at the clinical implications, when any of these systems fail, the consequences are just huge.
Oh, they're dramatic.
A classic respiratory one is COPD. The body gets so used to high CO2 that the central chemoreceptors they sort of give up. They lose sensitivity.
They become desensitized, yeah. So the body switches its primary stimulus. It starts relying almost entirely on that backup system.
The hypoxic drive. So suddenly, low oxygen is the only thing keeping the person breathing.
And that's exactly why giving high supplemental oxygen can be dangerous. You might accidentally remove their only remaining stimulus to breathe.
Wow. And then on the transport side, you have drugs designed specifically to target these pumps, like SGLT2 inhibitors.
For type 2 diabetes, yeah. They block that Na-glucose cotransporter in the kidney. So if you can't reabsorb the glucose,
You just excrete it and your blood sugar goes down,
simple as that. And conversely, you have a genetic failure like cystic fibrosis.
which is a defect in a chloride channel, the CFTR protein. It messes up water movement, and this tiny failure at the cellular level
Leads to that thick, sticky mucus throughout the entire body. Systemic consequences.
So when you pull it all together, the bottom line is the body is in a constant war against inertia. It's always regulating.
Always. Whether it's fighting a delayed signal in the brainstem or a steep concentration gradient across a membrane, it's all about maintaining equilibrium. And we saw how the core regulation of breathing can shift its entire dependency from CO2 to O2 because of chronic disease. So here's the final thought.
Okay.
If the sodium-potassium pump is the linchpin, the primary battery for the entire cell, what happens to the body's overall regulatory capacity when that ubiquitous foundational pump experiences even a minor chronic slowdown across billions of cells?
That's a sobering thought.
That's the true cost of entropy. Welcome to the deep dive.
Great to be here.
Today we're taking a look inside the body's own hidden chemical labs.
Specifically at how life manages these incredible chemical extremes.
Right. We're going to follow the journey of a few key ions, hydrogen, chloride, and bicarbonate, to see how they let us do two fundamental things: digest our food and, well, breathe.
And our mission really is to find the surprising link between them. The shared blueprint the body uses for both creating extreme acid and buffering massive amounts of waste gas.
It's a fantastic topic.
Yeah.
So let's start with the stomach. The environment in there is just hostile.
It's incredibly hostile. I mean, our sources are clear. Gastric juice can have a pH as low as 0.8.
0.8, wow. And the body's making what, up to 3 liters of this stuff a day?
Exactly. And it's not just acid. You've got pepsinogen, which becomes the enzyme that digests protein.
An intrinsic factor, which, if you don't have it, you can't absorb vitamin B12.
Okay.
That leads to pernicious anemia.
Right. So the big question is, how do these specialized cells, the parietal cells, produce an acid that strong without just dissolving themselves?
What's the core engine driving it?
The core engine is what's called the proton pump, the H plus K plus AT pace.
Yeah.
It actively shoves hydrogen ions out of the cell and into the stomach. But to do that, it needs a constant supply of them.
And where do they come from?
This is the clever part. The cell pulls in carbon dioxide from the blood.
Wait, CO2? Like the waste gas from respiration?
The very same. This is where the enzyme carbonic anhydrase comes in. It's ridiculously fast.
The fastest in the body, right?
It is. It grabs that CO2 and water, instantly makes carbonic acid, which then splits into a hydrogen ion for the stomach and a bicarbonate ion.
Ah, so that bicarbonate is the byproduct. So what happens to it?
It gets immediately pumped out of the cell into the blood in exchange for a chloride ion.
Which then follows the hydrogen ion into the stomach to make hydrochloric acid.
You've got it. And that southern rush of bicarbonate into the blood during digestion, that's what we call the alkaline tide.
The alkaline tide. So the blood actually becomes slightly more alkaline to balance out the acid being made?
For a short period, yes. It's a perfect balancing act.
Okay, so is that bicarbonate just treated like buffered waste, or is the body reusing it somewhere else? Let's pivot to the lungs.
Oh, it is absolutely reused. The system is far too efficient to waste it.
So let's talk about gas transport. We know how oxygen moves around mostly.
Right, 97% of it just binds to the iron in hemoglobin. The oxygen-hemoglobin dissociation curve shows it perfectly. It holds on tight in the lungs and then lets go easily in the tissues where oxygen is low.
Simple enough. But what about the CO2 waste? How does that get out?
Well, a little bit is dissolved in plasma, about 7%. Another 23% or so hitches a ride on hemoglobin.
But that leaves the lion's share. A massive 70%.
And that 70% has to be transported as the bicarbonate ion.
The same molecule.
The very same. And this is where we see that beautiful structural reuse. Inside the red blood cell, you find that same lightning-fast enzyme again.
Carbonic anhydrase.
Yep, it's right there, converting CO2 into bicarbonate.
So wait, is this the chloride shift? The red blood cell is using the exact same plumbing that the stomach cell used.
That is the ultimate connection. It's called the hamburger phenomenon, and yes.
No way.
The bicarbonate diffuses out of the red blood cell into the plasma in exchange for a chloride ion coming in. The remaining hydrogen ion is just buffered by the hemoglobin.
It's the same ion exchanger. That's just incredibly efficient design.
It's a single elegant toolkit applied to two very different, very extreme chemical problems.
And for you listening, this coordination really matters because when it fails, the consequences are serious.
Absolutely. If the stomach's acid production gets out of control, that's a direct path to peptic ulcers. The acid and pepsin literally start eroding the lining.
And on the flip side, with breathing, if you retain too much CO2, that bicarbonate system gets overwhelmed.
And you fall into what's called respiratory acidosis.
So to wrap up, the body is using the same core tools, carbonic anhydrous and this bicarbonate chloride swap, to manage everything from digesting a steak to running a marathon.
It's a masterclass in chemical logistics.
So we want to leave you with a final thought on just how fragile this elegant system can be.
Right.
Think about carbon monoxide, CO. It's an odorless, colorless gas.
And its attraction, its affinity for hemoglobin is just off the charts compared to oxygen.
It's 210 times greater, so it binds to hemoglobin and it just won't let go.
It forms carboxyhemoglobin, and your body's ability to deliver oxygen just collapses. It causes a massive left shift in that dissociation curve.
So think about that, how this entire beautifully synchronized life support system we've just discussed can be completely and silently dismantled by a single invisible molecule. Today, we're cutting right to the heart of how the body maintains, its internal balance. We're diving into two fundamental systems. The kidneys master water conservation, and the pancreas' incredibly sophisticated way of handling digestion.
Right, and what's fascinating is that both systems are constantly regulating these huge fluid volumes and some pretty intense chemical reactions.
Okay.
So our goal is to really get into the mechanics, the secret engineering that keeps your internal world running smoothly.
Okay, let's unpack this then. Let's start with the kidney and its amazing trick for not letting you pee yourself dry. The countercurrent mechanism.
Yeah.
The whole mission here is to establish this massive difference in salt concentration, a steep osmotic gradient in the renal medulla that can hit, 1,200 µmL?
An astonishing number, yeah. Think of it like creating this little hyper-salty ocean deep inside your kidney.
So how does it do that?
Well, you've got two structures working together. First is the countercurrent multiplier. That's the loop of Henle. The active ingredient here is what you could call a triple pump engine, the Nana plus AS plus Nita2Cl cone supporter. It's in the thick ascending limb.
Okay, the ascending limb.
It just pumps out all those solutes, but, and this is the key, that part of the tube is totally impermeable to water. So water can't follow the salt out.
So you're building up this huge salt pile outside the tubule.
Exactly. You get this massive salt buildup outside and incredibly dilute fluid left inside.
Okay, so the multiplier creates this enormous salt mountain, but blood is constantly flowing through the kidney. What keeps that gradient from just being immediately washed out?
That's a great question, and it's the critical role of the second part, the countercurrent exchanger.
The vasa recta.
The vasa recta. It's, a purely passive preservation system, and it follows that same U shape. Blood flows really slowly, and it just exchanges water and solutes, making sure that gradient stays intact.
So without the exchanger, the multiplier's hard work would be for nothing.
It would be undone in minutes, exactly.
And this whole mechanism, it defines the absolute minimum amount of water we need to survive, right? The obligatory volume of urine.
It's the non-negotiable cost of staying alive.
So if your body makes, say, 600 milliliters of waste a day, that has to go.
Right, and your kidney's max concentration is 1,200 millidose in a ml. Well, the math is fixed. You have to lose at least 500 millil, half a liter of water, period.
And if you disrupt that system,
then you see massive consequences. A loop diuretic, like furosemide, what does it do? It locks that exact triple pump engine.
It blocks the co-transporter.
Locks the co-transporter. And suddenly the kidney can't build that gradient, and the body just starts dumping huge volumes of dilute urine.
Wow, okay, that's an interesting story of conservation.
Yeah.
Now let's shift to the digestive system and look at fluid output. The pancreas, it's a powerhouse, right? Pouring out 1.0 to 1.5 liters of juice a day.
A huge amount. And it's a master multitasker with two critical jobs. First, its juice is highly alkaline. It's packed with bicarbonate.
So its first job is defense?
Purely defense. It has to neutralize that highly corrosive acidic chyme that's just been dumped out of the stomach. It protects the intestinal lining and, crucially, sets the stage for the second job.
Which is digestion.
Which is digestion. It has these powerful enzymes for everything. Fats, proteins, carbs, things like pancreatic lipase, trypsinogen.
And the trypsinogen is secreted in an inactive form, right?
A critical safety measure. They're like these little biological bombs with a delayed fuse. They only get activated once they are safely out of the pancreas.
Stop the organ from digesting itself.
Exactly.
The regulation is just, it's so sophisticated. It's a demand-based system, isn't it?
Absolutely. It's driven by two main hormones. When acidic chyme shows up, a hormone called secretin is released.
Okay.
Secretin's job is simple. It triggers a massive, watery, bicarbonate-rich flush. It's the neutralization crew.
So acid arrives, and the pancreas sends out the antacid.
You got it. But then, when the actual food products, the fats and proteins, arrive, a different hormone, CCK, is released.
And that one triggers the enzymes.
That one triggers a secretion that's super rich in all those digestive enzymes. It's resource efficiency perfected.
And when that goes wrong?
You see the clinical connection immediately. In diseases like cystic fibrosis, you have defective chloride transport, which messes up bicarbonate secretion. The fluid gets thick, sticky, clogs the ducts.
And the enzymes can't get out, leading to malabsorption.
Severe malabsorption, exactly.
So, okay, to sum this up, we've seen two highly complex systems. One is all about conserving water with these osmotic gradients. The other is managing digestion with carefully controlled alkalinity and this powerful enzyme cocktail.
Right, and it raises an important question, I think. We talked about that huge daily volume of pancreatic juice, 1 to 1.5 liters.
This incredibly alkaline fluid.
So what do you think the immediate and profound impact on the body's overall acid-base balance would be if the secretion of that highly alkaline fluid just suddenly stopped?
That's a huge metabolic shift to think about. A great question. Thank you for joining us for this deep dive, something to mull over. Welcome back to the deep dive. Today we're jumping straight into the heart's its electrical command center.
Right into it.
We're talking about how it generates its own rhythm, how it stabilizes blood pressure in an instant, and how we actually read that signature.
And it all starts with something that's, well, intentionally unstable. The heart's pacemaker, the SA node, it doesn't really have a resting state like other muscles.
No resting state. That sounds risky. If the goal is a steady rhythm, why would it be built on instability?
That's the key. It's because of these very unique channels. They're called the IF channels, but most people call them the funny channels.
The funny channels. Okay, I have to ask, why funny?
Because they're just always a little bit open. They're constantly leaking in positive ions, mostly sodium, so the cell can never actually rest. It's always, creeping up towards firing again.
So that's the baseline rhythm.
No.
But our heart rate changes all the time. So the autonomic nervous system must be in charge of that.
Exactly. The ANS doesn't turn it on or off. It just changes the slope of that climb.
The slope. So how fast it gets to the firing point.
Precisely. If you need to speed up, say you're exercising, the sympathetic system kicks in. Adrenaline. It steepens that slope by boosting those funny channels, and this is key, the L-type calcium channels.
So you hit the threshold faster, more beats per minute.
Much faster. And to slow down, the parasympathetic system, the vagus nerve, does the opposite.
It puts on the brakes.
Puts on the brakes hard. It releases acetylcholine, which flattens that slope way out. It opens potassium channels to pull the potential down and inhibits those calcium channels. It just takes a lot longer to fire.
And that's a clinical target, right? Those L-type calcium channels. I'm thinking of drugs like verapamil.
Yep. That's a huge mechanism for antiarrhythmics. You're directly blocking part of the ignition system to slow things down. It's a fine line to walk, though.
I can imagine. You want to slow it, not stop it.
Exactly.
Okay, so that's the heart's rhythm. What about the immediate second-by-second regulation of blood pressure, like when you stand up too fast?
That's a whole different and even faster system. That's the baroreceptor reflex, your body's instant buffer.
So baroreceptors?
Yeah.
They're pressure sensors. Where are they?
They're stretch receptors, really. In your big arteries, the carotid sinuses in your neck and the aortic arch, when your blood pressure shoots up, they get stretched.
And they fire off a signal?
A rapid fire signal straight to the medulla in the brainstem. The medulla immediately slams the brakes on the sympathetic system and fires up the parasympathetic.
The vagus nerve again?
The vagus nerve again. The result is instant. Your blood vessels dilate, lowering resistance, and your heart rate and contractility drop. BP comes right back down.
And without that, you'd get that head rush, the dizziness, orthostatic hypotension.
You'd probably faint every time you stood up. It's that critical.
Okay, so how do we see all this activity? The ECG, the electrocardiogram, it's the electrical map of all this.
Right, and it's really just a few key things. You start with the P wave, that's small. It's the atria depolarizing.
Then comes the big one, the QRS complex, that huge spike.
That's the ventricles firing. It has to be big and fast because they're so massive. We're talking under 1/10 of a second.
And then the T wave is just the ventricles resetting, repolarizing.
Exactly. But often the most important part isn't the waves themselves, but the flat line right after that QRS spike, the ST segment.
Right, because it's supposed to be flat, isoelectric. If it's elevated or depressed, that's a huge red flag.
A massive red flag. It's one of the clearest signs of myocardial ischemia or injury, a heart attack.
We should probably also mention the PR interval, the time between the P and the QRS, if that gets too long.
Yeah, over 0.20 seconds, that tells you there's a delay, a first-degree AV block. Conduction is being held up somewhere.
So the bottom line here seems to be that the heart is this beautiful system built on, well, controlled chaos.
It is. It's fundamentally unstable by design, but it's managed with these incredible lightning-fast feedback loops from the nervous system.
But here's a final thought, something to really chew on. You said the baroreceptors are instant lifesavers, but what happens in someone with chronic high blood pressure?
That's the fascinating and troubling part. Over time, they reset. They start to defend the higher pressure as the new normal.
So your body's fastest, most important short-term safety system essentially adapts to the dysfunction.
It does.
Then what does that mean for long-term control? If your primary reflex gives up and accepts the new dangerous normal, how much harder does that make treatment? And what slower, maybe hormonal system is forced to try and pick up the slack?
A very important question.
Something for you to mull over until our next deep dive.
Welcome back to the deep dive. Today we are digging into two just incredibly synchronized biological systems.
We're talking about the absolute mechanical precision of your heart and then the body's emergency patch kit blood clotting.
Exactly. You sent us some amazing sources on the lightning fast sequences that keep everything running, and our mission is to pull out those vital timed details for you.
And it really is all about timing, especially when you start with the pump, the heart.
Right. So the whole cardiac cycle, one beat, takes what? Less than a second?
At rest, yeah, it's about 0.8 seconds total, and you could split that pretty cleanly. Systole, the contraction, that's the short part, maybe 0.3 seconds.
And diastole, the relaxation and filling phase, gets the other 0.5 seconds.
It's so critical. But inside that 0.8 seconds, the sources pointed to these isovolumetric periods.
Yes. The isovolumetric contraction and relaxation, it feels counterintuitive because the blood volume inside the ventricle isn't changing at all.
All the valves are sealed shut.
So why are those moments so important?
Because that's where the most drastic pressure shifts are happening. During that first phase IDC, the ventricle is basically a closed box,
building up that huge force.
The force you need to blast open the aortic and pulmonary valves. Then, in IVR, the pressure just plummets, which allows the whole cycle to start again.
Okay, so speaking of filling up, let's talk volume. At the end of diastole, you've got about 120 milliliters of blood in there, the end diastolic volume.
Right, the EDV. But the key is, it doesn't all get ejected.
No, only about 70 milliliters, which is the stroke volume.
And you can actually hear this mechanical perfection, the classic lub-dub sound.
A heart sounds.
Exactly. The lub, that's S0. It marks the start of the squeeze of systole. It's the sound of the mitral and tricuspid valves snapping shut.
And the dub S euros must be the end of systole.
It is. That's the aortic and pulmonary valves closing. And that sound kicks off that critical 0.5 seconds of relaxation and filling time.
So here's the key question for you listening. What happens if you compress that timing?
Well, that's where things get dangerous. In severe tachycardia, say a heart rate over 180, your body robs time from diastole.
The filling phase.
The filling phase. So less time to fill means a lower EDV, which means a lower stroke volume. Your cardiac output just tanks. The synchronization is everything.
Okay, so if the heart's rhythm is the engine, what happens when the plumbing springs a leak?
Then the body deploys its rapid response team, blood coagulation. It looks complex, but it all drives toward one goal: a stable fibrin mesh.
A patch, and it starts in two main ways.
Two trigger pathways, yeah. The extrinsic pathway is the super fast one. Think major trauma. A chemical called tissue factor gets released, and it activates something called factor X almost instantly.
But what if it's just, damage inside the blood vessel? Exposed collagen or something without that massive tissue damage?
That's the intrinsic pathway. It's a bit slower because it has to go through a few more steps, factors 12, like horizon IX, but it gets to the same place.
Activating factor X.
Exactly. Both paths funnel into the common pathway. Once factor X is active, the main job is to convert prothrombin into its active form, thrombin.
And thrombin is the real workhorse here, isn't it? It takes fibrinogen, which is just floating around, and turns it into these sticky fibrin monomers.
Monomers that just spontaneously link up and form the clot. But, they can't do this efficiently without a little help.
There has to be something else.
Platelets.
Yeah.
They are absolutely essential. They provide this negatively charged surface that the whole activation complex needs to assemble on. They're basically the scaffolding for the repair job.
And just as important as starting this whole cascade has to be stopping it, right? How do you put the brakes on something designed to self-assemble so quickly?
The regulation has to be incredibly tight. You have molecules like antithrombin III that go around and shut down any excess thrombin.
Okay.
And then you have the protein C and protein S system, which is great at neutralizing some of the other active factors to make sure the clot stays local. It's all about that balance.
And understanding that balance is, it's why a drug like warfarin is so effective.
Exactly. Warfarin basically slows down the synthesis of several of these key factors that all depend on vitamin K.
Factors 2, 7, IX, and X, you're just tapping the brakes on the whole system to prevent dangerous clots.
So when you step back, you've got two incredible systems. One is a rhythmic mechanical pump driven by pressure, and the other is a chemical enzymatic cascade. The level of synchronous perfection is just staggering.
It really is. So here's a final thought for you to mull over. Considering both the heart cycle and the coagulation cascade are driven by these precise cascading reactions, what happens if the electrical events starting the heart become unsynchronized, while the chemical events starting a clot simultaneously go into overdrive? That combination, that's the edge of a biological disaster.
Welcome to the Deep Dive. Today, we're going really deep into the body's hidden genius.
Yeah, we are.
We're looking at these incredible mechanisms that manage compliance, control, and concentration.
Right. Lungs, bladder, kidneys. 3 totally different landscapes.
And we're using your source materials to figure out how these fundamental, background systems actually keep you alive.
That's the mission. We're trying to pull out those aha moments, those little molecular tricks and neural shortcuts the body uses.
And I want to see what happens when they go wrong.
Exactly. When these automatic processes break down, the consequences can be, pretty dramatic.
Okay, let's start with the lungs. Just the physics of keeping them open seems like a huge challenge.
It is.
We're talking about lung compliance, which is really just how easily your lungs can stretch.
And the biggest enemy here, according to the sources, is surface tension.
Right. Like trying to pull apart 2 wet pieces of glass.
Yeah.
That same force is trying to collapse all your tiny air sacs, the alveoli.
And the body's solution is basically a detergent. It's called pulmonary surfactant.
So what is it exactly?
It's mostly a phospholipid phosphatidylcholine, if you want to get technical made by your type 2 alveolar cells.
And its job is just to cut that tension.
Its only job is to get in between the water molecules, break up those bonds, and just slash the surface tension. It makes the lungs compliant.
Okay, but here's the really clever part I saw. Physics says small bubbles should collapse into bigger ones, right?
Laplace's law, P equals 2T over R. Yeah.
So why don't all our small alveoli just empty out and collapse?
Because the surfactant works better where it's needed most. It lowers the tension more in the smaller alveoli than in the bigger ones.
Ah, so it equalizes the pressure, it stabilizes everything.
Precisely. Without it, taking a single breath would be almost impossibly difficult.
Which brings us to the clinical side. Premature babies, born before 35 weeks.
They often haven't made enough of it. Their lungs are stiff, low compliance. That's respiratory distress syndrome.
Okay, so from air and fluid, let's move to, just fluid. Bladder control. Micturition.
Micturition.
And the sources are clear. At its core, this is a fully automatic spinal cord reflex.
All happening down in the cerebral region, S2 to S4.
Yep. But the loop is simple. The bladder wall, the detrusor muscle, stretches.
And that sends a signal.
That sends a signal to the spinal cord, and a parasympathetic signal comes right back, telling that muscle to squeeze.
But wait, if it starts squeezing, that's going to create even more stretch on the receptors, isn't it?
It absolutely does, and that's the whole point. It's a positive feedback twist.
So the contraction fuels an even stronger contraction.
Yes, until the bladder is completely empty. It's a self-reinforcing all-or-nothing reflex.
So what stops it from happening all the time?
Your brain. Your higher centers are constantly sending inhibitory signals down to keep that reflex in check.
So, what happens if you sever that connection, say in a spinal cord injury above S2?
You lose the voluntary control, but that local reflex is still perfectly intact. You get what's called an automatic bladder.
Meaning the moment the bladder fills to a certain point, it just empties. Suddenly, completely, and with no conscious control at all.
Okay, from controlled emptying to our last challenge.
Yeah.
Intense concentration.
The kidney.
The countercurrent mechanism.
Yeah.
This is just beautiful biological architecture.
It's all about how the loop of Henle and the vasa recta are arranged, right?
Right. You have the multiplier, which is the loop of Henle, and the exchanger, the vasa recta. They work together.
Okay, walk us through the multiplier. It sounds like the engine of the whole thing.
It is. The thick ascending limb is basically a salt pump. It actively pumps sodium, potassium, and chloride ions out into the kidney's medulla.
But, and this is the key bit, the wall of that tube won't let water follow.
Exactly. So you're pumping out all this salt, building this incredibly steep concentration gradient. You can get up to 1,200 million moles deep in the medulla, super salty.
And the exchanger, the vasa recta, is just there to make sure blood flow doesn't wash that gradient away. That's its job. The blood flows in the opposite direction, the countercurrent, so it can pick up salt on the way down and then lose it on the way up.
It preserves the gradient.
It perfectly preserves that hyperosmotic environment. Then the collecting duct passes through that salty region, ADH opens the water channels, and water just rushes out.
Creating concentrated urine.
Very concentrated urine, and if you lack ADH or your kidneys don't respond...
That's diabetes insipidus.
Right, you can't reabsorb that water. You get polyuria, massive amounts of very dilute urine, sometimes over 3 liters a day.
It's just incredible. When you pull it all together, you see how a single soapy molecule or a simple spinal loop, how they create these enormous physiological outcomes that we just take for granted.
It really shows how the body is constantly exploiting physics, surface tension, concentration gradients to keep things stable, and, you know, it makes you think. Well, we talked about how your brain has to actively suppress that bladder reflex. Think about all the other essential hidden reflexes your body has, and the sheer processing power it takes just to override them. That ability to consciously control our own internal autonomic world is, well, it's a huge part of what makes us human, isn't it? Okay, let's jump in. For today's deep dive, we're peering inside your body's control room. Our mission is to really explore the underlying systems that keep your internal environment, the milieu interior, so stable, you know, perfect, even when the outside world is throwing chaos your way.
What you're really looking at is the body's relentless, constant effort against entropy.
Entropy.
And that effort has a name. It's homeostasis. This constant maintenance of critical parameters.
Things like temperature, pH, blood sugar.
Exactly. All kept within a tiny optimal range. It's really the hidden, never-ending job of survival.
So if stability is the goal, how does the body enforce it? The primary mechanism we hear about is negative feedback. What's a good example of that in action?
Well, the most elegant part is how it automatically reverses the initial disturbance. So think about your body temperature rising. The control system kicks in, initiates sweating, widespread vasodilation.
You bring it right back down to that set point.
It's built to oppose any change. That's its whole purpose.
That opposition is the key. Although, you noted there's one critical exception, positive feedback.
Right, that's the system designed for a finish line. It amplifies the stimulus. The classic example, of course, is the accelerating cycle of oxytocin and contractions during childbirth. It just pushes the system forward and forward until it's done.
It sounds like a really high-stakes gamble. So how do we even measure the quality, the effectiveness of these regulatory systems?
We talk about the gain of a system.
Yeah.
And you can think of gain as the control system's ruthless efficiency rating.
An efficiency rating, I like that.
Yeah, you calculate the tiny error that remains after the system has tried to correct a problem. A very high score like The negative 100 you can see in some temperature control systems.
Wow.
It means the body's correcting nearly 100 times more than the tiny error it allows to linger. That level of efficiency is what keeps you alive.
It really makes you realize that the difference between health and catastrophe is just this tiny, perfect regulatory window. Because when that negative feedback fails, well, we see the results. Things like diabetes mellitus, where insulin regulation just breaks down.
Exactly. Or you can look at detrimental positive feedback, like in hemorrhagic shock. Falling blood pressure weakens the heart, which causes the pressure to fall even further. It's a self-sabotaging spiral the body can't pull out of.
Not without external help. So we've covered chemical balance. Now let's shift to how the body handles, physical command and layers of reflexes. We tend to think of reflexes as simple, but something like the defecation reflex, it's actually multi-layered.
It is. It's really two reflexes working in tandem. You have the weak intrinsic one that's run by the local gut nervous system, but the real power comes from the stronger parasympathetic defecation reflex.
And then that's the one that travels down the spinal cord.
That's the one. That powerful automatic signal travels through specific highway exits, S2, S3, and S4, to drive these really intense parasympathetic contractions.
What's so fascinating here is that even with this powerful automatic command, the final decision rests with the cerebral cortex. You, the listener, have that critical voluntary override over the external anal sphincter.
And that control is managed by the pudendal nerve. It's the highest center, literally overriding the lowest, most powerful reflex.
Okay, so let's pivot from that to the absolute extreme end of adaptation, moderate exercise. The body just throws every control lever it has. We see cardiac output spike.
Right, from higher heart rate, higher stroke volume.
But the true resource management skill here is how the blood is routed.
It's optimized delivery. It's beautiful, really. The body performs widespread vasoconstriction,
tightening the blood vessels
in all the non-essential areas like the gut and kidney, and combines that with intense vasodilation opening vessels, but only in the active skeletal muscles. All that resource gets delivered exactly where it's needed when it's needed.
That's phenomenal. And so if you subject your body to this repeatedly with endurance training, the system stops adapting just for the moment, right? It changes long term.
Yes. It leads to things like physiological cardiac hypertrophy, increased plasma volume, which is why you see the phenomenon of athletes' bradycardia.
Or resting heart rate sometimes below 50 beats per minute.
Exactly. And that's all due to an increased vagal tone.
Vagal tone, which is just a way of saying your body's main chill-out nerve, the vagus nerve, is now constantly whispering, slow down, to your heart. It shows the system adapting not just to survive, but to truly thrive.
From the sheer efficiency of those high-gain systems to the raw power of the S2, S4 parasympathetic reflex, the body is just this incredible machine built for constant internal stability.
So we close on this idea. If the body is a finely tuned machine, we've seen that the highest level of control is often voluntary inhibition. It's you holding the override button.
Which brings up a really important question to think about. What happens when the connection between the brain and that reflex center is severed? A spinal cord injury above those sacral segments can abolish the voluntary control you just mentioned. But the powerful automatic reflex below the injury, well, it may still remain active. Makes you wonder, how powerful are those automatic systems really when the conscious override is completely gone? Something to think about.
Okay, let's unpack this.
Do it.
We're diving into 3 systems that, you probably learned about ages ago, but we're looking for that high-level insight.
The aha moments, right? The bits that show just how ridiculously optimized the human body is.
Exactly. So our mission today is a rapid tour of 3 core defenses that keep you stable. We're talking chemical, mechanical, and then electrical.
It's really the ultimate lesson in stability. I mean, the body's entire existence is about controlling its internal boundaries.
So where should we start? What feels like the most basic system?
Let's start with what feels basic but is anything but simple: saliva.
Ah, okay. Nature's Swiss Army knife, you could say?
I love that framing, yeah.
Because everyone knows it's, 99% water, but that tiny little bit left over is pure function.
That's where all the work happens. You've got alpha amylase to start digesting food right away, but like you said, that's only half the story.
It's not just about food, is it?
Not at all. The other half is all defense. That fluid has lysozyme, which is an antibacterial enzyme, and then immunoglobulin A, IgA.
A major antibody. So it's a digestive aid, a chemical buffer, and a topical antibiotic, all at the same time.
Precisely. And what I found fascinating in the sources was the nervous system's control over it. It's like a switch.
The parasympathetic versus sympathetic response, right? So if you're just relaxing.
If you're chilling out, the parasympathetic system triggers this massive, watery, enzyme-rich flow. It's great for digestion, great for washing things away.
But if you hit the panic button, the stress response,
then sympathetic stimulation kicks in, and you get that smaller, thicker, mucus-rich flow. Which brings us right to the clinical hook.
xerostomia, chronic dry mouth.
Yeah, and it's not just annoying. If you lose that copious flow, you lose the buffering, you lose the washing action, and your risk for tooth decay just skyrockets.
The entire chemical defense just fails?
It fails.
Wow, that's a powerful demonstration. So moving from chemical defense, the body also needs a kind of emergency seal for when things go wrong.
Which brings us to hemostasis, the rapid controlled process to stop bleeding.
And the fascinating thing here is the contradiction, isn't it? How do you stop bleeding in one spot?
While keeping the rest of your blood flowing freely. It's a tightrope walk.
So it's a three-step process.
It is. First, you get the vascular spasm. The vessel just clamps down. Then the platelets rush in to form a plug. And finally, you get blood coagulation, the clot itself.
And the sources really highlighted the intrinsic pathway. For anyone who hasn't thought about this since, a college physiology class, it's like an internal tripwire.
That's a perfect way to put it. The trigger is so simple. Blood just has to contact exposed collagen from under the vessel lining.
And that one touch sets off a whole chain reaction.
A whole chemical cascade. It eventually activates something called factor 9, and that leads directly to the ultimate lock turner, factor 10.
So wait, if just one little key in that whole chain is missing, the entire seal just fails.
Precisely. The clearest example is hemophilia B, or Christmas disease. It's a deficiency in that one component, factor 9. It just shows you how critical every single link is.
Okay, here's where it gets really interesting for me. We've gone from a chemical defense to a mechanical seal. But how does the body communicate all this instantly?
The electrical system?
The resting membrane potential, RMP.
We're talking about the body's own battery, basically. The electrical difference across your cells, which is typically around negative 90 millivolts inside.
And the reason for this charge is surprisingly simple, isn't it? It's almost all about potassium.
It is. The cell membrane is just incredibly leaky to potassium ions, the K+ ions.
This is like this one-way door where positive potassium ions can just leak out much faster than anything else comes in.
Exactly. They leak out down their concentration gradient, and they leave behind all these non-diffusible negative ions. And that builds up the negative charge inside the cell.
But something has to maintain that steep gradient, right? Otherwise it would just balance out eventually.
Right. And that's the tireless sodium-potassium pump. The NAK pump.
The tiny bouncer.
The tiny, relentless bouncer. For every cycle, it actively kicks 3 sodium ions out and only brings 2 potassium ions in.
So there's always a net loss of 1 positive charge.
3 out, 2 in. That inequality means the pump itself is electrogenic. It actually contributes a little bit to that negative charge all on its own.
And the clinical side of this is compelling. The sources mention drugs like digitalis.
Yes, they inhibit this very pump in heart muscle cells. And through a chain reaction, that ultimately increases intracellular calcium, which boosts the force of your heart's contraction.
All rooted in that one tiny pump.
Incredible, isn't it?
Yeah.
So let's tie this all together. What does this all mean?
I mean, what we've seen are the body's tiered defense mechanisms. You have the immediate chemical buffer.
A mechanical clotting seal.
And then the fundamental electrical engine that makes everything else work. It's an absolute masterclass in biological defense.
It really is.
Yeah.
And to leave you with a thought on that electrical engine.
If that sodium-potassium pump works so hard to maintain order inside the cell, what happens when the external environment changes?
That is the crucial question. Consider hyperkalemia, abnormally high potassium outside the cell.
Right.
That external shift makes the resting membrane potential less negative. It pushes the cell closer to its firing threshold.
Which leads to hyperexcitability.
Dangerous hyperexcitability and cardiac arrhythmias. It shows how this delicate electrical foundation is so tightly wired that even slight fluctuations outside can short-circuit the whole system. Welcome back to the Deep Dive. Today we're doing a rapid-fire run through the body's most critical plumbing and pumping systems.
We're talking about the physics of it all.
Exactly. We're looking at glomerular filtration rate, that's GFR, and cardiac output, or CO. And I mean, the numbers here are just staggering. Your GFR is about 125 milliliters a minute.
Which sounds small, but do the math.
Right. It adds up to 180 liters of plasma filtered every single day. You are cleaning your entire plasma volume 60 times a day.
And at the same time, your heart is pushing out about 5 liters of blood every minute just at rest. And what's so fascinating is how these massive jobs depend on this incredibly precise balance of pressure and volume.
So that's our goal today.
Yeah, let's unpack the core mechanics. You know, how the body controls all this. It's a shortcut to seeing what happens when that balance fails.
All right, let's start with GFR. You called it the precise sieve.
That's what it is.
So filtration is basically the volume of plasma that gets pushed into this part of the kidney called Bowman's capsule. And it's passive.
Totally passive. The kidney itself doesn't spend energy on it. It's all driven by pressure.
Specifically, the glomerular capillary hydrostatic pressure, or PGC.
Exactly. That's the physical pushing pressure inside the kidney's own capillaries. The kidney gets that pressure from the heart, but it has this very, very clever filter to protect itself.
And that filter is what stops the big stuff from getting through, like proteins, albumin, for example.
Yes, but it's not just about size.
Hold on, so it's not just a physical sieve like a coffee filter?
No, not at all. The barrier has a negative electrical charge. It actively repels the negatively charged proteins like albumin. It's like two magnets pushing each other away.
Wow, so if that charge fails, you start losing vital protein. So how does the kidney keep that crucial pressure, that PGC, so stable?
Well, it mainly does it by tightening or loosening the little arteries that feed the filter, the efferent and efferent arterioles.
That's the autoregulation.
That's the day-to-day local control. But, you know, in an emergency, say, severe dehydration or hemorrhage, the body can just override that.
With hormones, like angiotensin II.
Right. Angiotensin-2 constricts everything to conserve fluid, and that actually lowers your GFR on purpose.
And that's a perfect link, actually, to the heart. Because angiotensin II is trying to raise the pressure the heart sees system-wide.
It connects everything.
So let's jump to the pump itself. Cardiac output. We said it's 5 liters a minute at rest, but it can scale up to, what, 35 liters a minute in an elite athlete?
An incredible range, and to understand that, you have to look at stroke volume, the amount of blood ejected with each single beat.
And 3 main things control that. First up is preload.
Preload is just how much the ventricle is stretched at the end of filling. Think of it like stretching a rubber band.
The more you stretch it, the harder it snaps back.
That's the Frank Starling mechanism, exactly. A fuller tank leads to a more forceful contraction.
Okay, so preload is how full the tank is. Then you have afterload. That's the resistance, right? The pressure it has to fight against.
That's right. Imagine having to push open a very heavy spring-loaded door with every single heartbeat. That resistance is the afterload.
So high blood pressure means a heavier door.
A much heavier door. And that makes it harder to push blood out, which can decrease your stroke volume.
And the last one is contractility,
which is just the muscle's raw intrinsic power, independent of the load. The sympathetic nervous system, your fight or flight response, can crank this way up.
Okay, let's unpack this then. We see the mechanics. What does it mean when things go wrong?
This is where it all comes together, because you really can't separate the two systems. A failure in one immediately affects the other.
So let's take the kidney first.
Okay, so when your GFR suddenly crashes, we call it acute kidney injury, or AKI. And the cause is often prerenal.
Meaning the problem's before the kidney. Not enough blood is getting there in the first place.
Precisely. Like in severe shock or dehydration, the perfusion pressure just bottoms out. Or the problem can be postrenal.
After the kidney. A blockage.
A kidney stone is a perfect example. The urine can't get out, so pressure backs up into Bowman's capsule and physically stops filtration.
And on the heart side,
you can have cardiogenic shock. This is when the heart itself fails as a pump, maybe from a massive heart attack. Your CO just plummets.
And if cardiac output plummets, there's no pressure to drive the GFR in the kidneys.
And that leads to hypoperfusion, not just in the kidney, but everywhere. The whole system starts to shut down.
So it's so clear, these life-sustaining functions are just this incredibly delicate dance of pressures and flows between the heart and the kidney. Okay, so here's something for you to mull over. We've established your entire plasma volume gets filtered 60 times a day through this incredibly precise barrier.
A barrier with a negative charge.
Exactly. So what happens if that negative charge is just slightly compromised? Not gone, just weakened. Proteins like albumin start to leak through. Is that lost protein just wasted, or does losing it into the urine trigger a whole new cascade of problems throughout the body? Welcome to the deep dive. We've gone through a huge stack of sources on what are
really the three pillars of how we function.
Right, how we build our internal transport system, how we defend it, and how we actually deliver the goods.
Exactly. So for this deep dive, our mission is to pull out the absolute need-to-know facts on erythropoiesis, immunity, and the oxyhemoglobin dissociation curve. We're going to give you the essentials.
Let's do it.
Okay, so let's unpack this and start at the beginning. The factory floor building red blood cells.
That factory is the red bone marrow. You find it mostly in flat bones, so, your vertebrae, sternum, ribs.
And this is where stem cells are just churned out into red blood cells.
And huge numbers. It's an incredibly efficient process. The most amazing part is probably the last step, where the cell literally kicks out its own nucleus.
Just to make more room for hemoglobin, for the cargo?
Exactly. It becomes a pure delivery vehicle.
But something has to control that. What's the master switch? And, where does it usually go wrong?
The master switch is a hormone, erythropoietin. Most people just call it EPO.
Right.
And it's produced almost entirely by the kidneys. The amazing part is the kidney acts like a tiny oxygen sensor.
So if oxygen levels in the tissues drop,
it notices, a condition we call hypoxia. The kidney then just cranks up EPO production, telling the bone marrow, hey, we need more delivery trucks.
And for that factory to run, it needs its raw materials. What are the absolute non-negotiables?
Well, for the replication part, for the DNA synthesis, you have to have vitamin B12 and folic acid. No way around it.
Okay.
But for the hemoglobin itself, the part that actually carries oxygen, the key ingredient is iron. And a lack of any of those is what leads to the most common types of anemia.
So if that EPO signal fails, you get anemia. But what if it goes into overdrive?
Then you get something called polycythemia. The blood literally gets too thick, which, as you can imagine, massively increases your risk of clots, of thrombosis.
Okay, so we've built the delivery truck. Now, how does the system defend itself? Let's shift to the security system. Immunity.
Right, and we can basically split our defenses into two main strategies. First, you have innate immunity.
That's the fast, non-specific stuff.
Exactly. Your skin, your natural killer cells, is the first line of defense. Then you have the second strategy, acquired immunity. This is the sophisticated, highly specific system.
The one that learns.
The one that learns, yeah. Mediated by lymphocytes. It gets smarter with every single encounter.
And when we talk about that specific defense, we have to talk about its main weapons.
We do. Those are the antibodies, or immunoglobulins. IGs for short.
I like to think of them as little guided missiles.
That's a perfect analogy. They're these Y-shaped proteins that are designed to find and tag a specific invader for destruction.
Now, there are 5 classes, but a couple of them have these really surprising, almost paradoxical jobs.
Yeah.
Let's talk about IgG and IgE.
Sure. So IgD is the most common one, but its most unique trick is that it's the only one that can cross the placenta.
So it gives the fetus immunity from the mother. A biological inheritance.
It is, a head start. And then you have IgE on the other side of the coin. It's meant to protect us from things like parasites, but it's also the one responsible for freaking out.
You mean allergies?
Allergies, asthma. It's the system overreacting in the most spectacular way.
Okay, and here's where it gets really interesting for me. The final step, the actual delivery. The oxyhemoglobin dissociation curve, the ODC.
That classic S-shaped curve.
Right, and that shape tells us that binding is cooperative. Once one oxygen molecule binds, it makes it easier for the next one.
It does. And we measure hemoglobin's affinity for oxygen, its stickiness, with a value called P50.
Which is normally about 26.6 millimeters of mercury.
Correct. But the most critical idea here is the right shift. That just means hemoglobin's affinity for oxygen goes down. It gets less sticky.
So it can unload the oxygen more easily into the tissues that are screaming for it.
You got it. It's the body being smart, adjusting on the fly.
So what causes that rate shift? What are the signals that tell it to drop the cargo?
It's all part of what we call the Bohr effect. There are 4 big ones: increased CO2, increased acid, so a lower pH.
Increased temperature.
And, this is a big one, increased 2,3-DPGG, a little molecule inside the red blood cell that basically acts like a crowbar, prying the oxygen off hemoglobin.
And the opposite, a left shift where it holds on too tight, is a real problem. We see that with stored blood, right?
We do. It's called the blood storage lesion. The banked blood loses that 233-DPG crowbar over time, so when you transfuse it, it's not very good at releasing oxygen, at least at first.
So, tying this all together, what's the bigger picture?
The bigger picture is that it's all one beautifully interconnected homeostatic loop. You have the kidney controlling the bone marrow from afar with EPO.
And then at the local level, you have CO2 and acid directly telling the red blood cells exactly where to make the delivery.
It's just elegant macro and micro control working together.
It really is.
Yeah.
Which leaves a final provocative thought for you to chew on. We know severe acidosis causes a strong right shift, which is great for getting oxygen to tissues. But if oxygen pressure in the body gets too low, that same right shift can actually make it harder to load oxygen back in the lungs. So how does the body balance the urgent need to release oxygen in the tissues with the absolute need to reload it in the lungs?
That's the billion-dollar question.
Something to think about until our next deep dive.